Hello friends, welcome to Agriculture channel. I am Varsha Gaitonde and this video is about tissue culture basics. As you know, tissue culture refers to the technique of growing plant cells, tissues, organs, seeds or other plant parts in a sterile environment on a nutrient medium. Culture media used for in vitro cultivation of plant cells are composed of the basic components like first and foremost is complex mixtures of salts that consist of essential elements or mineral ions, organic supplements like vitamins or amino acids, then carbon sources, usually this is a sugar or sucrose, gelling agents, plant growth regulators, antibiotics, complex mixtures of salts. These include essential elements of mineral or mineral ions important for plant nutrition and their physiological functions. The essential elements can further be divided into two categories, that is, one is macroelement and another is microelement, and third one is iron sources. First of all, let us see the role of growth culture. Whenever we want to grow something, there should be some basic media. If we are cultivating something in field, soil acts as a basic media. If we are cultivating something like aeroponic or hydroponic, there is also something used as a base. Growth media provides water, it provides mineral nutrition needs, vitamins, growth regulators, access to atmosphere for gas exchange, and removal of plant metabolite wastes. We'll discuss about the macronutrients one by one. The major macronutrient is nitrogen. As you know, NPK are major nutrients for any of the plant growth. Nitrogen influences growth rate, essential in plant nucleic acid, protein, chlorophyll, amino acids and hormones. Phosphorus is abundant in meristematic and fast growing tissues, which is essential in photosynthesis and respiration. Potassium, it is necessary for cell division, meristematic tissue, helps in the pathways for carbohydrate, protein and chlorophyll synthesis. This also provides disease resistance to plants. Next is calcium, which is involved in the formation of cell walls and roots and leaf development, participates in translocation of sugars, amino acids and ties up oxalic acid. Means this helps in get rid of the toxic compounds. Next is iron. This is involved in respiration, chlorophyll synthesis and photosynthesis. Iron, sodium EDTA. This sequesters iron, making it available to plants. Next is magnesium. This is involved in photosynthetic and respiratory system, active in uptake of the phosphate and translocation of phosphate and starches. Coming to the macronutrient salts. Here I have enlisted the major macronutrient salts, and those are ammonium nitrate, potassium nitrate, calcium chloride, magnesium sulfide, potassium hypophosphate, iron or sodium ethylene diamine tetraacetate, that is EDTA, boric acid, manganese sulfate, zinc sulfate, then potassium iodide, sodium molybdate, cupric sulfate, cobaltous sulfide. Coming to the macronutrient salts, the first and foremost is sulfur, which is involved in the formation of nodules and chlorophyll synthesis. Here gives structural components of amino acids and enzymes. Manganese, this is involved in regulation of enzymes and growth hormones, assists in photosynthesis and respiration. Molybdenum is involved in enzymatic reduction of nitrates to ammonia. This assists in conversion of inorganic phosphate to organic form. Zinc is involved in the production of growth hormones and chlorophyll, active in respiration and carbohydrate synthesis. Boron is involved in production of growth hormones and chlorophyll. This is active in respiration and carbohydrate synthesis. Copper is involved in photosynthetic and respiratory system. This assists in chlorophyll synthesis and used as reaction catalyst. Coming to the organic compounds, first one is the carbon source like sucrose, which may be sometime glucose or fructose. Then vitamins, first and foremost is adenine, that's a part of RNA and DNA. Then inositol, which is a part of B complex in phosphate form in part of cell membrane. Organelle and is not essential to growth but beneficial. Then thiamine, which is essential as a coenzyme in the citric acid cycle. Next group is organic acids like citric acid, which is required 150 mg per liter, typically used with ascorbic acid like 100 mg per liter as an antioxidant. This can also be used some of the Krebs cycle acids. Next is phenolic compound like chloroglucinol, which stimulates rooting of shoot suctions. Coming to the natural complexes, coconut endosperm, fish emulsion, protein hydrolysates, tomato juice, yeast extract, malt extract, potato agar are some of the natural complexes utilized in tissue culture media. The next component is charcoal. Activated charcoal is used as a detoxifying agent. Detoxifies waste from plant tissues. Impurities and absorption quality vary. The concentration is normally 0.3% or lower. Charcoal in tissue culture helps in acid washing and neutralizing, but it can never be reused. Coming to the agar. Agar is nothing but a gelling agent. There are two things always we get confused. One is agar and one is agarose. Agar is a gelling agent that is used in tissue culture, and agarose is again a gelling agent or media that is used in gel electrophoresis, or where marker technology is used, agarose is used. Coming to the discussion about agar, it's a gelatinous substance derived from the seaweeds. It's an unbranched high molecular weight polysaccharide obtained from the cell walls of some species of the red algae, primarily from the genera Giladium and Gracilaria. Agar is insoluble in cold water but forms relatively inert gel that melts at about 100 degrees Celsius and solidifies at around 45 degrees Celsius. Using agar as a gelling agent, main advantage is that agar does not react with any components of the medium and it is not digested by enzymes from the plant tissue. If necessary, agar can be washed to remove inhibitory impurities. Agarose is a purified extract of agar without the agaropectin function and sulfate groups. It is required in lesser quantities since it has a higher gelling strength. Agarose is used for more demanding procedures like culturing chloroplasts. Consists of AD123 galactopyranose and 3,6 anhydro-Al-1,4 galactopyranose linked into polymer chains of 20 to 160 monosaccharides units. Gallon gum, also known commercially as Phytagel or Gelrite or Gelwell, is used primarily as gelling agent alternative to agar in microbiological culture. It's able to withstand 120 degrees Celsius heat, making it especially useful in culturing the thermophilic organisms. One need only approximately half the amount of gallon gum as agar to react, and equivalent gel strength through the exact texture and quality depends on the concentration concentration of divalent cations present. Gallon gum is used as gelling agent in plant cell culture on petri dishes as it provides a very clear gel facilitating light microscopical analysis of the cells and tissues. Coming to the growth regulators, first and foremost is auxin that is required for root development, cytokinin for shoots, gibberellins for cell elongation, ascorbic acid for plant stress hormone, then ethylene which is not required, but we will discuss one by one. There is certain things like hormonal combinations like callus development, adventitious embryogenesis, rooting of shoot cuttings, then adventitious shoot and root formation. The important auxin components used in tissue culture is IAA, that is indole 3-acetic acid, indole 3-butyric acid, naphthalene acetic acid, 2,4-D, 2,4,5-T, CPA, NOA, MCPA, then dicamba, pecloram, etc. Important cytokinins are 6-benzyl aminopurine, then benzyl adenine, 2-IPA, or we can say 2-isopentyl adenine, then DPU, kinetin, zeatin, then didiazuron. Here we will see the important characters of gibberellin, abscisic acid and ethylene. Gibberellin is not generally used in tissue culture. Gibberellin tends to suppress root formation and adventitious embryo formation, whereas abscisic acid primarily a growth inhibitor but enables more normal development of embryos, both zygotic and adventitious, whereas ethylene, the question is not how much to add but how to get rid of it in vitro. Natural substance produced by tissue culture at a fairly high level, especially when cells are under stress, that will produce ethylene, and ethylene enhances senescence. That's why ethylene should be kept away from the tissue culture media. It suppresses embryogenesis and development in general. Coming to the different tissue culture medias, the first and foremost is Morishige and Skog medium. This is used for micropropagation, organ culture, callus culture and cell suspension culture. In 1962, they prepared this medium for in vitro callus culture of Nicotiana tabacum, belongs to family Solanaceae. Second is banana medium. This is used for in vitro micropropagation of Musa family. Banana medium is the modification of Morishige and Skog 1962, which provides all essential elements to grow banana in vitro. Next important media is Gamberg's 5 medium. This is used for the callus culture and cell suspension culture. This medium is established by Gamberg in 1968 for callus and cell suspension culture of glycine max, which belongs to family Fabaceae. This medium is widely used for in vitro cell, tissue and organ culture. Remember, Morishige and Skog is required for micropropagation, organ culture, callus culture, cell suspension culture. Banana medium for growth of banana, in vitro micropropagation. Then Gamberg's 5 for in vitro plant, tissue and organ culture. Especially this is used for glycine max. Next medium is niche medium. As I have already told, the amount of nutrients required are really low, like White's medium, and here glycine and other components are also used. This medium is used for anther callus culture. This is established by Niche in 1969 for in vitro anther culture of Nicotiana. Head breadth medium or SH medium. This was developed by Schenk and Hinderbrand in 1972, used for callus and cell suspension culture. Then CHU or Chu medium. Chu medium was developed by Chu and co-workers in 1975. This is used to promote organ culture and cell suspension culture. It is useful for generating new useful genetic varieties in Gramineaceae family or plants by the initiation, growth and differentiation of callus from rice pollen culture. Remember, this is utilized in rice pollen culture. Next is BM medium. This is used for seed culture and micropropagation of orchids. Remember, BM medium is mainly used for orchid seed propagation. It was developed by BM or Van Vass in 1986 for in vitro cultivation of protocombs from orchid seeds. We will discuss about the different medias. In different medias, the macronutrients, as I already discussed, and their quantity is different in different medias, and similarly the micronutrients, it is given in the tabular form. Important vitamins used are thiamine, pyridoxine, nicotinic acid and myoinositol. Here the range of concentration varies and the range is very important. The amount is mg per liter. Thiamine, maybe the amount varies from 0.01 in White's media to 10 in Gamber's media. Similarly, pyridoxine again 0.01 to 1 in Gamber's media and 0.01 in White's media. Nicotinic acid 0.05 in White's media and 1.5 in niche media. Then myoinositol, that is in constant concentration, that is 100. Here you should remember, in White's media the amount of organic supplements used are quite low. Other components used like glycine, that varies from 2 to 3. Then folic acid concentration is 0.5 and biotin 0.05, that is only used in niche media, and pH of the media should be 5.8. Remember this point. Coming to the major steps in tissue culture, first one is the initiation phase. In this, the first phase of tissue culture, where the interest is obtained and introduced and sterilized in order to prevent any microorganisms from negatively affecting the process. It is during the stage of the tissue it is initiated in the culture. Second is multiplication phase. Here the in vitro plant material is re-derived and then introduced into the medium. Hence, medium is composed of appropriate components of growth, including regulators and nutrients. Remember, always in the initiation phase, a proper tissue is taken from any plant. It is excised, it is sterilized and kept ready for the initiation. Then in the second stage, multiplication phase, it is supplied with adequate amount of whatever the components growth media is required, and because of that media it will start multiplicating the cells. This step is often repeated several times in order to obtain the desired number of plants, and this is the main stage where contamination occurs. Stage 3 is root initiation, where hormones are required in order to induce rooting, consequently complete plantlets. After rooting, shooting will happen, then hardening. After hardening, the plant will be transferred to the field condition. Coming to the procedure of tissue culture, for this media preparation is required, maintaining the proper balance of pH, dissolving all the nutrients in adequate quantity or prescribed quantity, sterilization to avoid contamination are the major steps in order to get the free disease-free plants. Here some steps are given how the plants are prepared and how they are treated. Thank you. I hope you liked this video. If you liked, please do not forget to hit the like, comment and subscribe the channel. Thanks once again.